Hello, this is the Naked Mole Rat podcast by Libo Studio. I'm Ilya Kolmanovsky, and we're running a bi-weekly show in Russian about scientific discovery and the people behind it. I talk to a lot of the world-rate experts, so most of these interviews are in English. So we thought it makes sense to give the international audience a chance to listen to them. The Naked Mole Rat. It lives long, digs deep, and serves the science. On this episode, we've got a very special guest, Professor Michael Levin from Tufts University. He has created an entire field. He publishes up to 50 papers a year and leads a high-profile Paul Allen Center, which was created specifically to support Levin's research. And here's why. Levin has invented a new way to think about the cells which build our body. He claims that cells have minds. He can actually read their thoughts, hack into their minds, and manipulate development. His experiments can lead frogs to grow new limbs, eyes on their bellies, or turn frogs' skin into swarms of biorobots.
Yeah, good morning. This is Michael Levin. I'm a professor of biology. I work at Tufts University in Boston, and my background is computer science and biology.
So why don't we start with the following? For me, it's a kind of a simple textbook picture, the way I see the world. Everyone who listens to this podcast right now started her life with one cell. And we're all different. And usually we explain this by just saying we've got different genes, different DNA. For you, how did it start? How did your quest begin? And what's lacking in this simple picture of the world?
Wow. Well, there's a lot of questions there. How it started for me is simultaneously along two lines. When I was very young, I remember, and I'm sure there were other things that I don't remember, but what I do remember are two systems that I was looking at. One was at one point, my father showed me the inside of a TV set, and it was one of these ancient Russian television sets with the vacuum tubes and everything. We took off the back and we were looking in there, and it sort of it hit me, this amazing fact that someone knew how to put all those parts in there to get the correct outcome at the other end. So this idea that there was this hardware that you can arrange in a particular way, and then some sort of energy flows through it, let's say electricity, and some very interesting things then can happen. And so it was clear that the distribution of that hardware was essential for making those things happen, but that actually an important part of this was not in the hardware itself, because that same device was able to produce all sorts of different, TV shows and whatever. And the information wasn't actually in the hardware. The hardware was a machine that was constraining and enabling this energy to sort of manifest in various behaviors. So that was super interesting to me, that distinction between the energy and the physical machine. At the same time, I spent a lot of time outside looking at bugs and insects, and I started to think about what the situation there was. First of all, do we have the same kind of thing, the same distinction between software and hardware? What might the software be? How might it be? How might it relate to the physical structure of these critters? And how did they get there, right? So in the case of the TV, we know someone put it together very rationally and sort of had planning and whatnot. How do these creatures come to be? And especially the fact that they self-assemble from an egg, they self-assemble into these amazing complex creatures. So kind of that interplay between that, the physics of it and the biology, I think is to this day how I think about these things. And the other question you asked, which is what's missing in this basic picture, I think that for a lot of people, and certainly people in the field who think about these things, they know that they know better, they know the distinction. But a lot of people, when they think about the properties of specific plants and animals, they'll say, well, it's in the genes, right? And this statement of it's in the genes is actually very tricky, because we now know what's in the genes. So what's actually in the genes are protein sequences. And so when you're talking about anything other than protein sequences, including shape, behavior, the symmetry type of an organism, the ability to regenerate, all these interesting things, there's a huge gap between the protein sequences and this large scale system level behavior, which is often computational and so on. And so it's very tricky to say something is in the genes, because what the genes do is they specify your hardware. The genes are telling you what proteins, include ion channels, various other things that every cell gets to have. But the next step is to actually understand how that machine enables the behavior that we see, and that's the interesting part.
The behavior being self-assembly or growing a shape?
Yeah, both. So living things execute problem-solving behavior in multiple spaces. So they do it first of all, in physiological space, right? So every cell has to handle its various homeostatic needs and metabolic needs and so on. So there's physiological space. Then there's morphospace, so the space of all possible configurations of an organism. So cells have to work together to find the correct region of morphospace that corresponds to a fish, a frog, whatever it's going to be. And then there's the three-dimensional behavior space after that. So then it starts moving around, and so I see all of life as solving problems in these various spaces using some of the same tricks to navigate all of them. But that's
And what are these?
Yeah, so that's a very interesting question. So navigating efficiency these spaces is, to me, the definition of intelligence. And this is why there's a whole spectrum of intelligence, because some of these tricks are extremely simple. So, for example, bacteria have this thing called run and tumble, where they basically, right, the algorithm is very simple. If life is getting better, keep going in the direction you've been going. And if suddenly your situation now is worse than it was a few minutes ago in terms of food concentration or whatever, then randomly turn to a different direction and start running that way. That's a strategy. So that's a pretty good strategy. Bacteria make very good use of it, but it's a very simple one, and it's purely local, and it uses very short memory. It uses no or limited predictive look ahead. That's a very simple strategy. Lots of different other biological systems have much more complex strategies, going all the way up to human level and presumably beyond, where you can have things like metacognition, where you actually know what you know, and you can represent counterfactuals, and you have all kinds of complexity. But there's a whole spectrum in there with all sorts of interesting strategies of different sophistication for getting from wherever you are in these spaces to wherever you want to be. So the ideal set point.
I know that you collaborate with a philosopher, Daniel Dennett.
Yeah.
Why do you need a philosopher to do your job? Why do you need this collaboration for what you pursue?
Yeah. Why do we need a philosopher to do this job? Well, I guess the first question is what is the job, right? So I see my job as several different things. Certainly basic discovery is the key. And the important thing about having a philosopher, and I think actually Dan Dennett was the one who said this, that when you think you're doing philosophy-free science, you're not. What you're doing is science with a whole bunch of philosophical assumptions that you've just taken on board without prior examination. I don't believe it's possible to do science without philosophy at all. So you need some sort of philosophical perspective on what you're doing and what it means. In particular, the collaboration with Dan is very interesting because he is, of course, a leading figure in thinking about minds and cognition. And that underlies my whole view on biology, all the way from basic questions of evolution and things like this, all the way up to very applied biomedical and regenerative medicine applications that we're doing. I think there's a new field emerging at the intersection of computer science, biology, and cognitive science. And that last part, that cognitive science, the fact that most of these systems are agents in an important way, they're decision making, they have memories, they have preferences, and so on, is really key to regenerative medicine. So it has practical applications that we can talk about, but foundationally, trying to understand what is mind and how different kinds of minds can be embodied in different types of bodies, really in unconventional architectures, things that not brains, things that we wouldn't normally recognize as a mind. This is really important. And working with someone like Dan, and I work with other philosophers as well, but working with someone like Dan is really critical to have that kind of input and that history of thought about these things in ways that biologists are just now starting to intersect with.
So are you saying that cells have minds?
Yes. Oh, sure. Yeah. I mean, let's understand what we mean. Okay. So one really important thing about everything that I'm going to talk about, the perspective here, is that there are no binary categories. So I really reject this binary notion where you can look at something and you can say it has a mind or it doesn't have a mind, right? I think that's fundamentally wrong, and that gets us in trouble in many ways. I think that all of the interesting properties here, so cognition, minds, all of that, are spectra. They're a continuum, and so the question isn't whether something has a mind. The question is how much, and it might, and what kind. And it might be very little, and it might be almost zero, or it might be quite a lot. But yeah, absolutely, to properly understand cells, and even more so to understand how cells cooperate and collaborate towards large-scale goals like building an organ or regenerating something and so on. All of that requires us to understand what kind of mind and how complex and what is it capable of these collectives have. Yeah.
So given the body of the experiments that you have performed over your career, could you please give us a tour of the Umwelt of a cell? Imagine a cell sets out to build an extremity, a paw. So what's going on in its mind? Yeah, so I should preface that by saying that most of my work is not about the minds of individual cells. We've done a little bit of that, but other people have done much more. And so what I would talk about is the Umwelt and the mind of a cellular collective. And so first of all, let's just back up for a second and point out that all cognitive agents are made of parts. We are all, you and I both feel like we have centralized goals and memories, and we have preferences and past events that belong to us and so on. But we're a bag of cells, right? Really, and all cognitive agents are, whether or not you're an AI or a living creature, you're made of parts. And so the key mystery in all of this, maybe the most profound mystery of all, is how do large numbers of small competent agents work together to create something that's much larger than themselves, that has goals and memories that doesn't belong to any individual one. So your memories don't belong to any one neuron, they belong to you, but what are you? You're a collection of parts. And so there's this really important field called collective intelligence, which looks at how cognition scales up. How do you get these larger agents? So when you have cells that are working together to build something like a limb or an eye or whatever, you can certainly ask about the cognition of individual cells, and it will be, modest things like knowing what the local environment around them is like and having some sort of memory as far as what kind of signals they received in the past from nearby cells. They have preferences in terms of they will move towards specific substances and they will move away from other substances. You have a little bit of ability to, even yeast and bacteria have a little bit of ability to predict forward what's going to happen based on past experience. So if you pulse at a specific time frame, you pulse certain impulses, it will catch on to that pattern. Even cells can do that, and so on. So certainly there are very local intelligences within those cells that are handling very local goals, like keeping in a metabolic frame, appropriate set point. In other words, to have the right amount of nutrients and these kinds of things. But where the action really is for development is the mind of the collective agent. So one way to think about it is as the mind of an anthill. So, you have anthill. Mm-hmm. Anthill, yeah. So you have colonies of ants and termites, and these colonies know things that the individual ants don't know. So there's all kinds, and so there's lots of people, studying this in various ways. So the exact same thing is going on in development or regeneration. You have a collection of cells, which together, it's the mind of the collective that remembers how to build specific things. It's not the individual cells. And it's the collective that decides when it's enough and when they should stop building and when the correct limb has formed. No individual cell knows how many fingers you have, but the cellular collective absolutely does. And it will stop when the correct number of fingers is finished. And so the kinds of things that cellular collectives concern themselves with are goals that are anatomical. They're goals in this kind of space of all possible configurations that they could make. How does heredity work? Why does Xenopus look like its parent Xenopus? Yeah, that's a very interesting question. Why does a Xenopus look like its parent Xenopus?
because and in fact it doesn't have to, and so that's one of the most interesting things that I think that we found so far is this incredible plasticity, and so I'll get to that momentarily. What happens when it doesn't look like its parent? But the standard story is basically this: that what the DNA does is specify the cellular level hardware that every cell has, and what evolution has done is shape that hardware to have a very special property. That property is all other things being equal, meaning if you don't do anything else to it, the default case, you just leave it alone, it self-assembles into a very specific thing pretty reliably, right? So most embryos, have probably less than 1% error. And so what the DNA does is it specifies this incredible hardware that when left in a standard environment, always produces the same kind of outcome.
It was broadcasts program Время as in your childhood.
Yeah, I mean more like I would say it's somewhere between that and the test pattern. Those color the thing that came on at 11 p.m. or whatever, the color that color test pattern. It's something that happens by default. That's called the species specific default morphology. That's just what happens naturally. But the really interesting thing is that that's not the only thing that can happen. And cells, in fact, are perfectly willing to build other things, even with the same hardware. And how you know that is, first of all, very, very old experiments on chimerism, meaning mixing things, and you can mix genetically, so you can have a horse and a donkey and you can make a mule. So that's interesting. Even the genetics can be intermixed. And so given this weird genome, the cells actually know what to do, and they make something, right? It's neither a horse nor a donkey, but it's something. And then you can also mix cells. So you can take cells from different species and you can put them together, and they also work together. They're interoperable, and they will again make something. So what our work on this, the main work that we've discovered is basically in two lines. One is the latest thing we call Xenobots, and we call them Xenobots because they are created from the skin cells of a frog that is called Xenopus laevis. And so what we do is we take and this is in collaboration with some computer scientists at the University of Vermont, Josh Bongard. What we've done is to take the skin cells from a frog embryo and basically liberate them from the context of the rest of the embryo, take them out. And so Doug Blackiston, who's a staff scientist in our group, does this, and he takes these cells and he puts them in a different environment. And what these cells will do in the absence of the rest of the embryo is they will build something else. They build a little creature. It's a spherical little thing that drives around underwater. It has mobility on its own, so it has all kinds of moving behaviors. It has the ability to heal itself after damage. They cooperate with each other to build to rearrange their environment in specific ways. All kinds of interesting behaviors. It doesn't look anything like a tadpole. It doesn't look anything like a frog. What it does is repurpose its genomically derived hardware, all the metabolic machinery, the communication machinery, the cilia, the little hairs on the outside that it uses to sort of swim against the water. And it repurposes all of that towards a completely different anatomy. Now, what's interesting, I've given this talk where I showed 20 minutes of these things moving around and swimming and doing things, and then I say, what do you think this is? And people in the audience will say, wow, it's you got it out of a pond somewhere, it's some creature. And I say, well, we've sequenced the genome. And say, oh, that's great. Now we'll get to the bottom of this. And of course, the genome was 100% Xenopus laevis. It's frog, and you don't see any of this from the genome. And so that underlies this amazing plasticity of these cells. That there's a default behavior, but they're actually very competent to do other things in other circumstances, right? So that's one kind of one thing we've done. And what's really important is to think about the limitation of today's understanding of all of this. Despite all the progress in molecular biology and genetics, imagine a very simple thing, and this is something that we're actually doing. You can make an embryo we call a frogolotl. So this is 50% frog cells, 50% axolotl cells, right? Axolotls are these Mexican salamanders. Now, baby axolotls have legs. Baby frogs do not have legs. They're tadpoles. So if you mix 50-50 cells, will it have legs? I mean, you have perfect access to the frog genome. You have perfect access to the axolotl genome. Can you tell whether this thing will have legs? And the answer is no. We have no idea. I mean, we're working on this now, but you would think that this is the kind of question that we should be able to answer, and we can't, because while we have really good understanding of the hardware, we still don't have a very good understanding of the software that makes decisions about whether or not you're going to have legs and what kind of legs you're going to have. So understanding the hardware is really not sufficient. So that's the one line of research. The other line has to do with planaria. And so what we found is that there's an electrical circuit that flatworms, these planarian flatworms, use for a very interesting purpose. When you chop them into pieces, every piece will regrow whatever is missing to be a perfect worm. So you can cut a worm into lots of pieces, and every piece makes heads and tails and what they need to be a perfect worm. And so there's an electrical circuit that we discovered that dictates where the head and where the tail is going to go, and thus sets the number of heads and tails. And what we found is that if you temporarily rewrite the information that's present in that circuit, you can cause the pieces to make, let's say, two-headed worms. Now, the amazing thing is that circuit has memory, meaning it keeps whatever information you put in there. So once you make the two-headed worms, they stay two-headed. So in the future, if you cut them again, they can once again make two-headed worms. And so that, despite the fact that they have perfectly wild-type genomes, there's no genomic editing in this process. There's no transgenes. There's nothing wrong with the genomic sequence. And so that information, how many heads you are going to have, isn't specifically readable from the genome. So there you see this divergence.
You started to mention electricity. Could you please talk a bit more about it, the role of bioelectricity? Maybe an example of the frog with an eye on its belly could help us here.
Sure.
Could you please tell us about it?
One way to start thinking about how do cellular collectives work together to form complex objects, such as eyes and limbs and so on, you can start to think about, well, how do cells communicate? How do we put together machines that have memories and can do complex things? Now, we use electricity for our computational devices. The brain uses electricity to store goals and to orchestrate these neurons into a central intelligence. And so it turns out that the ability of the brain to do this, all these cool tricks that brains do, didn't just appear magically when brains evolved. Evolution found this very early on. So even bacterial biofilms use electricity to compute and to spread information. Basically, evolution discovered long ago that electricity is just a really good way. It's a really good way to exploit physics to integrate information across distance, between different cells and all that. So what people have found, and the first discoveries on this were like prior to 1900 even, but lots of people have been doing some of this work. And my lab in particular made some of the first molecular tools to do this. All cells, not just your neurons in your brain, but all cells communicate electrically. So all cells form these kinds of electrical networks, all cells in the body, and they form these electrical networks. And then you can use all of the tools of neuroscience and machine learning and artificial intelligence and all of that to ask, okay, if you have an electrical network made of cells, what does that network do? What are the memories? What are the information processing tricks that it can do? And so what these electrical networks do is they store pattern memories. They store information that's used to create anatomy. And the reason we know that is two types of experiments. First of all, you can use the various techniques to image those electrical conversations that cells are having with each other. You can see the maps. You can look at now we can look at there. What we do is we use these voltage reporting chemicals. They're basically fluorescent dyes that light up in different ways that you can then just see on a microscope, and they report the local voltage level. So you can take a frog embryo and you can soak it in this dye, and then you just use a microscope and immediately you see, we have these videos. It's quite incredible, actually. You see these videos of all the electrical conversations that the cells are having with each other. It's a little bit like imaging the brain. There are people doing neural decoding where they try to image the electrical activity of your brain, and from that decode what the information content is. So that patient is thinking about a flower pot or whatever. And so we do the same thing. We image the electrical patterns in developing embryos, in regenerating limbs, in tumors, in all kinds of things. And we try to decode those patterns to see what it is to really try to recover the information content of what that network is doing. What are the tissues thinking about, literally? So that's all well and good, but that's just kind of an observational step. The really critical step is the functional step, where you try to change those electrical patterns. And when you change those electrical patterns, the information that that collective is using to guide its anatomical growth and development and so on, that's the information. And so when you change that information, the cells do other things. And so one very early application was we noticed that there's a particular electrical pattern in the face that causes the formation of an eye. And so we asked the simple question, what if you remove that pattern, meaning change the electrical activity so that that pattern is no longer there? And what happens is then you don't get an eye. But even better than that, you can reproduce that pattern somewhere else in the embryo. And so what we did was we reproduced that electrical pattern in the gut, or you can do it on the tail, or it doesn't matter. You can do it anywhere, anywhere else in a frog. This is all, of course, frog embryos. So you can do this in another location in the frog embryo. And
how do you do that?
And so how do you do it? So there's a few different ways to do it. One way to do it is, so let's just remember where these electrical patterns come from. So the electrical patterns are set by a set of tiny proteins called ion channels, and they sit in the membrane of the cell, and they can open and close. When they open and close, they pass charged molecules, things like chloride and sodium and potassium. They pass these charged ions in and out. And by passing charges in and out, they change the voltage that's inside the cell. This is like standard neuroscience, basically, that when your neuron fires, it's because particular ions came in and are out, right? That changes the voltage of the cell. So we can do exactly the same thing. We can open or close these channels in any cell, or we can introduce new channels. This is what we developed almost 20 years ago, are some of these technologies to change the electrical signals by manipulating the ion channels that these cells have. And all cells have ion channels, but we can even give them new ion channels by injecting RNA that codes for these channels. And so the trick is just to figure out which channel do you want to give you the correct voltage change. You know, if I want to say this cell is normally at 60 millivolts, I'd like it to be 30 millivolts. Well, then what you can do is to take out some you can put in some potassium or take out some chloride, and you can calculate all of that. What do you need to do? So what we did in that case, in the frog case, is we took a particular potassium channel RNA, we injected it into cells in some other portion of the embryo, let's say cells that are normally going to be gut. And then, sure enough, it produced the electrical pattern specific to an eye. And guess what the cells built? They built an eye. So you can have an eye on the tail of a tadpole. Now, the couple of cool things about that. First of all, the animals can see out of those kind of eyes. So that's amazing. Yeah, there's plasticity. The brain has no trouble adjusting to the fact that, hey, my visual information is now coming from my tail. It's fine. The brain can do that. So that's incredible.
But what about the nerve?
Yeah, in that case, what happens is the optic nerve comes out of that eye. It starts to look around where it should go. It doesn't find the brain. Often it finds the spinal cord. Good enough. It connects to the spinal cord. That information somehow gets loaded onto the spinal cord and eventually makes its way to the brain. And the animals can see. We could test that with behavioral visual learning studies. And the other amazing thing, of course, is that we didn't have to specify all the information that's required to actually build an eye. I mean, think about it. There are I don't know how many cell types in an eye, right? And normally, if you're a bioengineer thinking of making an eye, you think, wow, I have to say, well, here's where the retinal cells go, and there's a bunch of layers, and then here's the cornea and the lens and the optic nerve and all this stuff. We don't know how to do that. Yeah, right. We don't know how to do that, but that's okay, because we took advantage of the system software. The system software has a really cool feature. It has a subroutine called Make an Eye. And the way to trigger that subroutine is with a very particular electrical signal. But like all subroutine calls, they're much less complex than the actual subroutine. So calling that module, that eye-making module, is a simple signal. It's a particular electrical pattern. But once you've triggered it, all the other stuff that is needed to make an eye, all the changes in gene expression and cell movement, everything else, all that stuff kickstarts. So for regenerative medicine, you realize that, wow, what that means is we can trigger complex anatomical features that we really don't know how to micromanage, and that we won't be able to micromanage, not in our lifetime, I don't think, but triggering them, I think we absolutely can. And so that takes advantage of the native software of the system, and that kind of modularity is really powerful.
Fascinating. Mike, I don't have a single more question. You've covered it so beautifully. Thank you so much.
Thank you so much. You too.
Take care.
Yeah. Bye-bye.
This was an interview with Michael Levin, a development biologist from Tufts University, as a part of Naked Mole Rat podcast by Libo Studio. I'm Ilya Kolmanovsky. Thanks for listening.